I took the figure from the WHO 4th edition book. Shows us the important steps of the B cell development. So we have two main compartments: central lymphoid tissue, which is bone marrow, and we have peripheral lymphoid tissue, which is lymph node. So this precursor B cells or progenitor B cells, very first B cells, it's also called B lymphoblast, produced in the bone marrow, and then they become a pre-B cell and then immature B cells. So these immature B cells exit from the bone marrow. These are called naive B cells. They can either be exposed by an antigen. So when they expose to antigen, there are basically a few ways they can go. They can become a short-lived plasma cells, or they can enter into the germinal center, or they can go directly and become a memory B cells or marginal zone B cells. So this germinal center stage is important. They first become a centroblast. So this area we know as a dark zone, which mostly composed of centroblasts, and they can become a centrocyte, which is also called cleaved cells, and this area is also called light zone. And you can see in the normal follicles, you can separate the dark zone and the light zone, because there are also some other cells, for example, tangible body macrophages, area that has high turnover. You can also see this high turnover in the Ki67 or MIB1 immunostain. This that stain will also show you the dark zone and the light zone separation. Centrocytes either can die or they can become exit the germinal center and the follicles, and they can become a long-lived plasma cells, and they can produce different types of immunoglobulins on their surface. They can become memory B cells or marginal zone B cells. So this two way of becoming a memory B cells and marginal zone B cells is important, because this in the germinal center there is something called somatic hypermutation occurs, and this hypermutation of the immunoglobulin genes is very important, especially for some of the lymphomas. Let's give one example. For example, chronic lymphocytic leukemia, small lymphocytic lymphoma arise from these memory B cells and marginal zone B cells, and they can arise from the cells that either goes through this process, and they called it mutated, like hypermutation occurs in these cells, or they can arise from directly from this cell, which not goes into the germinal center, and these are called unmutated cells. So why is this important? Because unmutated CLL type has more aggressive disease course compared to mutated ones. So okay, we have another cells like this big octopus-like cells which antigens on their surface. This is called follicular dendritic cells, also called FDCs. So this is important for antigen presenting. This is an important antigen presenting cell. We can use CD21 immunostains to see if there's a follicular dendritic cell meshwork. So this compartmentalization is important to understand first how this B lymphocyte development occurs, and the other aspect is understand some of the lymphomas that arise from these cells. B ALL, B acute lymphoblastic leukemia arise from these precursor B cells, and in the progerminal center cells, which is also called naive B cells, can become a mantle cell lymphoma. And in the germinal center, like centroblast and centrocytes can produce follicular lymphoma, which we know different grades of follicular lymphoma. It depends on the number of the centroblasts and centrocytes. Also, Burkitt lymphoma can arise from germinal center cells. Diffuse large B-cell lymphomas or Hodgkin lymphomas can arise from here, which these are usually CD10 positive lymphomas. So for the post-germinal center neoplasms, those can be marginal zone lymphomas arise from here. As I already discussed, can be CLL, SLL, arise from either these two cells. They can be lymphoplasmacytic lymphoma or diffuse large B-cell lymphoma, or plasmacytoma, because plasma cells also in the is considered post-germinal center cells. So we covered some of the basics of the B lymphocyte produced in the bone marrow and goes into circulation and goes into lymph nodes, peripheral tissues, and become a different type of cells, goes into somatic hypermutation and become as plasma cells or marginal zone B cells, and some of the important lymphomas that arise from these compartments.